Welcome, everyone. My name is Eleni Perraki. I'm a senior product manager at CentoCustom, and today, together with Jorge Pinto, Vice President Medical Genetics and a clinical geneticist, we will be sharing with you our new CentoCustom Panel, a new approach in diagnostic panel testing, and we would be also very happy to answer questions. Our agenda for today would be an introduction to the medical need for flexibility in panel testing, then to show you Centogene's solution in how we are bringing the pieces together. We will talk about the clinical utility of this solution, and at the end, we will answer any questions you might have. And please feel free to use the chat for the questions in the meantime. So, welcome, George, and over to you.
Hey, Eleni, thank you very much. Uh, Welcome, everyone. So the goal of today's webinar is to talk about the utility of gene panels currently in medical practice, and also how we can use gene panels in a slightly different way by customizing them to allow for additional flexibility, but also doing it in a way that is not too complex. So the primary use of gene panels still today, and in this era of genome sequencing and exome sequencing, NGS panels, please, still have a role. The main goal of when to use a gene panel is to maximize clinical sensitivity, meaning bringing together, testing at the same time, a group of genes that are known to cause disorders that have overlapping clinical features, but at the same time, reducing or even sometimes excluding incidental findings, secondary findings, and variants of unknown significance that sometimes are more difficult to deal with in the clinical practice. So when is an NGS panel still a preferred approach? Well, in situations or in the clinical context of a group of disorders that has very similar and overlapping clinical features. Sometimes also depending on the local context, because reimbursement is possible for panels, but not for other types of tests like exomes and genomes. When a clinician prefers to focus only on a specific group of genes to minimize the risk of incidental findings and to reduce the chances of having a result variant of unknown significance, or also because there are either best practice guidelines or local testing guidelines that are or that prefer that only a predefined set of genes is tested. The issues or the challenges with pre-designed panels, so panels that are already available to be requested, is that they may sometimes not meet exactly the needs of a specific patient or a specific clinic or a specific local context. Either, once again, because there are an SOP or a guideline recommending the test of a specific group of genes, and the pre-designed panel does not conform to that recommendation, or because there is a new gene that has been published and might fit the clinical picture of our patient, and therefore is not available to be tested in the context of a panel. Or we have a very nice but very, very large panel that we prefer to focus the analysis on just a subset of those genes, or also because we are also engaged in clinical research and we want to have a suspicion of a particular gene that is a candidate gene that could be involved in a specific disorder, and we want to test all our patients with a similar phenotype, for that gene in the context, but still in the context of diagnostic testing. And also because most panels available from all genetic laboratories are exome-based, if there is a need to include genes that are either non-coding or to explore non-coding regions of the gene, using a different backbone, like for example, a whole genome, might be relevant, might be helpful, might increase the diagnostic yields. Also, when we are comparing, and specifically as a clinician, when we have a patient and we want to select a panel, probably we won't find two panels exactly with the same exact gene content. And sometimes that is challenging because comparing different panels from different providers is not easy. So if we want to keep the same panel for all our patients, probably going with a pre-designed panel is not the best choice. Also, because the number of genes that is associated with disease every year is around 250, 300 per year, it's very difficult to keep any panel perfectly updated. So it could be useful that when the clinician has a patient or a group of patients and knows about a new gene that has been associated with disease and wants to include it in their panel, then it's possible if you can customize it, but not if it's a predefined fixed set of diseases, of genes. And now I'll hand over to Eleni to show us the our new offering, the CentoCustom Panel, and how that may benefit the clinical practice.
Thank you,
Up to you.
George. So, CentoCustom Panel is a solution that can bring all the missing pieces of panel testing together. So, the aim of this solution is to empower physicians with the maximum flexibility to be able to create a panel that is tailored to individual patient needs. So, CentoCustom Panel, it's available via our ordering portal, and with simple steps and in an easy ordering process, it allows full flexibility in diagnostic panel design. So we offer the flexibility to choose the backbone of the panel between WES and WGS, so between our exome, our centoxome, and our centogenome backbones. We allow full flexibility in the gene selection, so a panel creation can be from 2 to more than 4,000 genes from CentoGene's complete diagnostic gene list, but also we allow an additional option of adding research genes in the panel in case this is something of interest. And of course, the panel analysis is backed by CentoGene's biodata bank of more than 1 million patient cases that we have analyzed. And of course, the analysis is done by our experts, and of course, our expertise in diagnostic gene and variant curation, and also medical interpretation of the results. The turnaround time of this test is 15 business days. So with this new tool, we are placing the patient at the center of the diagnostic process. We are allowing a creation of a patient-centric approach in diagnostic panel testing. So who should consider ordering this test? This test is for physicians that would like to have a targeted approach for specific clinical cases, or that they would like to limit the analysis and the interpretation of results to specific aspects of their patient's clinical phenotype. It's for clinicians and geneticists that would like to have control over the gene selection and the ability to include the latest gene discoveries in their diagnostic panel. It could also be used from researchers and research teams that are interested to go beyond the diagnostic gene list in their analysis, and of course healthcare providers that are looking for flexibility in diagnostic testing. There are different ways of starting the panel customization. And so the different options could be to, in order to select a gene list, could be to start from a predefined panel and modify it. A predefined panel could be one of CentoGene's pre-existing panels, or a already customized and saved panel in one's account. And you can use this as a starting point to add or remove genes. The second option would be to use a phenotype-based design, so to use specific patient symptoms or specific disorders as filters in order to create a gene panel. You can upload a gene list from your computer that you have saved, or copy and paste a gene list, or you can cherry pick from our complete gene list. We have 4 different panel sizes: small, medium, large, and extra large, depending on the number of genes. So the ordering process, as I mentioned already, happens via our portal. It's a very easy process. So it's starting by deciding the design option, then the backbone selection between exome and genome. You can use the different filters we have in order to customize the panel, in order to end up with your gene list, which can include diagnostic genes and also research genes, and the panel is ready for ordering. To start the design process, the system will ask you to decide which is the preferred option of creating a panel. So to start from the complete gene list, or to use one of the existing panels, or to load and paste a list of genes, and then decide on the backbone. So this is not restricting at all, because the additional filters can be used at any time, and also the backbone can switch at any time if during the design process you would decide that this is that the preferred backbone is not exome, for example, but genome, or vice versa. So this is our CentoCustom Panel interface. And what you can see here is the table with the genes. Then you can see the different filters for creating a gene list. You can see the basket where all the genes can be seen, and we have 3 different view options. So you have all genes, you have diagnostic genes, and research genes. And then at the bottom of the basket, you can see the total genes there. The system will calculate the panel category depending on the size, meaning the amount of genes that are in the basket. And of course, there are additional options to include raw data in the analysis, and to save the panel under a preferred, in your account, under a preferred panel name that can also be shown on the medical report. So now let's talk a little bit more about the diagnostic gene list. So here you see how the gene list of the like the table of the gene list is shown. We include comprehensive information in order to be able to, in order to provide to our customers all the information they need to decide on which backbone they would like to use. So you can see we have agency symbol for each gene, agency ID, the disease association. We have a column called GDR. This means gene disease relationship, and it defines whether a gene is diagnostic or research. We have performance information for exome and genome, and the coverage of the gene on the 2 different backbones, on exome and then genome. And performance is calculated using different parameters, not only the coverage, but also, for example, if the gene contains homology regions, or if the main variants associated with this gene are not detectable via NGS analysis. And then you see we have a column called Additional Features CentoGenome, and I will explain this in the next slides, how by using the genome as the backbone, additional features are available for certain genes. Now, in order to be able to allow also the research gene option, what you would have to do would be to switch on the button where it says show all genes, and then the system will show all the genes, diagnostic and research. And in order to understand which ones are diagnostic and which research, you can use the GDR column. Now, in order to add genes in the gene list, this is very simple. You would just need to add the button add, and then the gene would be present in the basket, let's say in our gene list. And of course, you can change the view to see all genes or to see only the diagnostic genes or only the research genes. And here you can also see information about which backbone is selected. Here you see CentoGenome. In case the backbone, in case someone is interested in changing the backbone, this can happen at any time. So, as I mentioned, we allow backbone selection. This means that someone can decide between genome sequencing or exome sequencing. So, of course, when we're talking about exome sequencing, we are focusing on the coding regions of the genome, whereas genome sequencing, it's sequencing of the whole genome. But also, exome sequencing, also there are some differences in terms of the different type of variants that can be detected between exome and genome sequencing. So with exome sequencing, we can detect single nucleotide variants, small insertions and deletions, copy number variants, and mitochondrial variants, mitochondrial DNA variants. With CentoGenome, we can detect all the variants that can be detected with exome sequencing, but additionally, we can also detect additional structural variants and some more variants that I will talk about in the next slide. So what is the advantage of using genome as the backbone of a panel? So first of all, we can go beyond the coding region. This means that intronic variants can be part of the analysis. It also means that we can include non-coding genes as part of the analysis, which is an emerging field in recent medical and scientific advances. It also means that with genome sequencing, we can have more uniformity in the coverage of different regions. And also, we are enabling the ability to detect more complex variants. And of course, our genome sequencing also includes specific algorithms that can detect variants that are associated with important disorders. And this means that this can minimize the need for complementary assays. And to talk a little bit more about that, so using genome sequencing as a backbone, it can enable repeat expansion screening for certain neurological repeat expansion disorders. It enables spinal muscular atrophy CNV analysis for SMN1 gene, and we can also detect variants that are conversion between GBA1 and its pseudogene and are associated with Gaucher disease and with Parkinson's disease. Now, to show you how this would be seen on the tool, so how would the complex variant detection be seen? This is exactly the additional features of CentoGenome. So for example, on the top part of the page, you can see FMR1, which is a gene that is associated with repeat expansion variants. And you can see as additional features of our CentoGenome that it's mentioned repeat expansion screening. So if the backbone of the panel is genome sequencing, and this is part of the gene list, then by default, repeat expansion screening will be part of the analysis of the panel. And one other thing to mention here is that you also see the performance difference between exome and genome, between centoxome and centogenome backbones for this gene. And you see that it's not because of the coverage of the gene, it's exactly because of the repeat expansion variants. So for exome, we know that we have this limitation. We cannot detect repeat expansion variants, and this is why the gene has limited performance on centoxome. However, with centogenome, we are able to detect repeat expansion variants, and this is why the performance of the gene is high. And then on the lower bottom of the page, you can see another example, a gene RNU2-2. This is a non-coding RNA gene, and you can see that in centoxome, it is not available. This is exactly because it's a non-coding gene. However, however, in centogenome, the gene is available and the performance is high. So in a genome-based panel, this gene can be part of the analysis. Now let's talk a little bit about the different filtering options that can be used in order to shortlist or to create a gene list for a diagnostic panel. So we have filter by symptoms, filter by panels, upload a gene list, and load an existing panel. So one second. Can we have the full screen view? Thank you. So let's talk about the symptom-based approach. So here, when we filter by symptom, then we have the ability of using a symptom of our patient. So we can start typing a term. For example, here we are using muscular as the example term, and then the system will return all the symptoms that can be found with this using this term, and also the genes that are associated with this symptom. Now you can start selecting the symptoms that are associated with your patient's phenotype, and then apply the filter, and the gene list will be shortlisted to only show the genes that are associated with your patient's exact phenotype. The same approach can be used, but for a disease-based focused approach. So again, with the same tool, the system will also give you the option of returning the diseases that are associated with this term, and you can include all the genes that are associated with the suspected diseases in your panel for this patient. Now, the second filter that can be used is to use one of Centogene's existing panels. Here, you can filter by panels and start typing the panel you're interested in, or also pick from the whole panel list of Centogene. For example, here as an example, I have used CentoCancer, and you see that on the top of the page, you see how we have started typing CentoCancer. The system returns all the available options, and we have picked CentoCancer Comprehensive, and you can see the difference in the number of the genes from the top, from 25,000. Choosing CentoCancer Comprehensive, now the gene list has 118 genes, which are the genes that are part of this panel. You can decide either to add all the genes in your panel list or to choose the genes you want. An additional filtering option is to upload a gene list you already have. This can be done either by uploading a file or via copy and pasting a list of genes, and then the system will shortlist the gene list only, including the genes you have selected. And lastly, you can use as a starting point one of the previously saved panels in your account. And so once you choose this, the table will only show the genes that are part of this panel, and you can decide if you want to add more genes, remove some genes, or maybe use the exact same panel for additional patients. And now I will go back to George, that he will talk to us about the importance of gene curation.
Thank you, Eleni. So at Centogene, we use the CleanGen framework to evaluate gene disease association, meaning how well is it established that a variant in a pathogenic variant in a specific gene cause a phenotype, cause a disease. And the reason for that is that not only because we have a variant in a specific gene that is necessarily disease causing, even if it's predicted or we actually know that impacts the gene product, right? So there, for diagnostics, it's really important to have the correct evaluation of the gene disease association, so we can interpret correctly the significance, the clinical impact of that variant for a specific patient. Not only that, but also it has an impact in classification, the variant classification, because especially for gene disease associations that are only, let's say, limited, the maximum classification that a variant should have should be no higher than a variant of unknown significance. Therefore, this is key for when we are selecting genes, either for diagnostics or if we want to look at them from a clinical research perspective. We actually use this framework during our regular routine exome, genome, and panel testing. Every time we come up with a variant in a gene, that's either to select genes for panel design, or when we come up with a variant in a specific gene in an exome or a genome that is not or has been evaluated a long time ago, or it's new. And we use also our internal biodata bank with cases that have variants in that specific gene to complement the assessment of published cases and other reports that we may find in the literature. Actually, we have published this a couple of years ago when we started this activity of a systematic gene disease relationship curation. And also, we had the additional benefit of either establishing or improving the strength of 61 pairs gene disease association. In total, up to now, we have evaluated more than 700 genes and phenotypes, so gene phenotype pairs, during our routine analysis. When you select and order a custom panel, we will proceed the following way. So all the diagnostic genes will be evaluated as, let's say, a regular NGS panel. We will review and classify the variants, and we'll report on them using the same standards as we use for the pre-designed panels. So with comprehensive evaluation from our clinical scientists and also the medical interpretation by our medical team. And after the report is issued, you still get the same lifelong commitment that we provide, that anytime a variant that was reported is reclassified, you will receive a reclassification report with the updated classification for that specific variant on the one hand, and also the impact for that specific patient, either if a diagnosis is confirmed or excluded, or whatever it applies to that specific case. This is, let's say, a snippet of a report, of a custom panel report, and what is different from let's say, our normal pre-designed NGS panels is that here you have, of course, not only the indication that it is a custom panel, but also the backbone that was used. And if you decide if you want to have a custom name for your panel, you can add it during the request, the test request, and we will add it here so you can clearly identify the panel you designed. So you can even add a version. Let's imagine you have work in a clinic that is dedicated to a specific group of disorders. You can define your own panel that you apply to all patients for that specific clinic, and you have a consistent application of testing for the same genes for similar patients. Then you have, as always, the pictogram and the clinical interpretation. Then you also have in the appendix all the genes that you have selected and any technical limitation that may apply to any of the genes. Regarding the research variants, it's quite similar in the sense that you also have a table with all the variants in research genes that pass all the filters that we established that are described here in the section, and also you have a list of the research genes that you have selected and any technical limitation that may apply to one or more of those genes. So what is, let's say, in the clinical practice, how can patients benefit from this? So first of all, it's a different way of selecting genes for testing. So you can adjust the gene list to a specific patient, to the specific clinical picture of a patient, or if you prefer to have a consistent testing strategy for all patients in your department with a specific group of clinical features or clinical suspicion, you can have a consistent, customized, tailor-made panel. Either because you want to restrict the genes that are tested because of you want to focus on those that for which there is a treatment guideline, or because you have a specific project in clinical research where you want to focus the analysis in said gene list. This actually has important application in oncology, where in many contexts, either regional or even by guidelines, the recommendation is to not go for very broad genetic panels, but also focus in specific genes that are appropriate for the clinical context of a patient. Also, you have here the flexibility to go beyond the regular sequence variants and copy number variants. If you have a patient or work in a specific specialty, medical specialty that requires additional testing, for example, in the context of ataxias, where repeat expansions are a very important type of variant that explain a significant number of cases, and you want to include in the single test the repeat expansion screening, then you can go with the genome backbone and then proceed to testing your patient or patients using your set of genes that you have defined, and also include this different type of variants. Or once again, if you want non-protein coding genes and that you want to add to your gene list, you're more than welcome to do so, and we will of course analyze if the gene is diagnostic, we'll analyze it in the context of the clinical information provided. And a few examples in the last couple of years. So last year, RNU 42 was described, beginning of the year. Then later on, RNU 22, and then in the meantime, like 3 or 4 more genes of this, let's say, family of genes have been described. And if we wait for all the panels to be updated, we for sure will be missing. We are not going to provide our patients with the most up-to-date gene list. So once you are aware that a new gene has been associated with the disease that is relevant for the patient or for your work, you can immediately add it to your panel and customize it accordingly. And also in clinical research, so you may have the need, or you may want to, in addition to a set of diagnostic genes, to further explore other genes that either still don't have a gene disease association or established, or it's still very early days for that association to be fully confirmed, and you may want to test your patients to see if you have a consistent segregation of not segregation, a consistent appearance of relevant variants in those genes with the phenotype that you are searching for. So I hope that we were able to show you that this approach is a simple approach in the sense that you can very easily customize your tests, your panels, either for your patient, for a specific patient, for a specific group of diseases that you may be interested in, and also to support your clinical research, because in the end, what we all want is to maximize the diagnostic yield and provide patients with answers and with firm diagnosis.
I would really like to thank you, George, for presenting today and sharing your expertise on this topic. And thank you all participants for your attention. And please feel free to email us for any questions or to arrange a demo.
Thank you, Eleni. Thank you, everyone.
Have a nice evening.